Okay, welcome everybody. Welcome to the special seminar. It's our great honor to welcome Professor Stephen Chu from Stanford University to the physics department this afternoon. Professor Chu has had a remarkable life and career filled with scientific accomplishment and public service at the highest levels. And in the interest of leaving a little bit of time for his actual talk, I'm going to keep this introduction as brief as possible. So he completed his PhD in physics at Berkeley and then went on to Bell Labs, where he began the work on laser cooling and trapping of atoms, for which he would go on to win the Nobel Prize in Physics in 1997. After Bell Labs, he moved to Stanford University as a faculty member and also served as department chair twice, I think. And then moved to LBL, Lawrence Berkeley Lab, in 2004, where he served as director of LBL, until he was tapped in 2009 by President Obama to serve as Secretary of Energy in President Obama's first administration, which he did from 2009 until 2013. After that, he returned to Stanford, where he is both a professor of physics and professor of molecular and cellular physiology in the School of Medicine. In addition to the Nobel Prize, he's won many other awards and honors, and I will list just a few of them. He's a member of the National Academy, the American Association for Arts and Sciences, the American Philosophical Society, Academica Sinica, the Chinese Academy of Sciences, the foreign affiliate of the Royal Society, the Korean Academy of Science and Technology. The list goes on. He's received literally dozens of honorary degrees from universities around the world. So with that, it's my pleasure to introduce Professor Chu. Please take it away.
Okay, so I'm going to talk about entropy, molecular motors, and statistical physics. It's biology, so I'm sympathetic to all the physicists in the audience. Are there any biologists or biophysicists? Please say, okay, great. Okay, so I'm going to start with the second law of thermodynamics. And the second law of thermodynamics, this is according to Rudolf Clausius. And it said heat can never pass from a colder to a warmer body without some other change connected therewith occurring at the same time. What he really meant is heat goes from hot to cold and not from cold to hot. And to get heat to go from cold to hot, you have to do something, like run a refrigerator. So that was what he was saying. This second law holds a special place among physicists. And Albert Einstein, in his autobiographical notes in 1946, said classical thermodynamics is the only physical theory of universal content that will never be overthrown. That the law that entropy always increases, the second law of thermodynamics, holds, I think, the supreme position among the laws of nature. If your pet theory of the universe is against the second law of thermodynamics, I can give you no hope. There is nothing to collapse in deepest humiliation, said Arthur Eddington. And finally, a very short quote that says, for the second law of thermodynamics, I will burn at the stake. Okay, so it was pretty special. The primary leaders started with Sadi Carnot way back when the conservation of energy was not even known. A young military person who did something really amazing, but the people who put it on a firm physical basis was Clausius and Thomson, Lord Kelvin. And so what did Sadi Carnot think about or do? And so for that, I'll remind you of what it is if you are able to do things in a frictionless way. Suppose you want to climb a mountain, and you get to start and you go to the finish, and then you go from the top of the mountain back down. Because it's frictionless, the energy going from path 1, 2, or 3, all these paths over here, and let's see if this, no pointer. So let me just see, does that pointer show up? Yes. Barely, okay. So if it's frictionless, you can go up path 3, go down path 1, go up path 2, go down path 3. Any round trip means no change in energy. If you go slowly and start slowly, no kinetic energy. And so then you say the potential energy is conserved. So what did Carnot think about? He was trying to explain what are the limits on a steam engine. But a steam engine was water and it was condensing, and he says, that's too complicated. So he said, let's not talk about water condensing. Let's just talk about some idealized type of gas. And what he said is, suppose you put a piston with some gas in it onto a hot temperature, and you let the piston expand very, very slowly. So the heat on the hot temperature then collides with the gas molecules, but it's expanding very slowly. All those collisions are elastic. So it says, okay, great. So you can transfer all the heat. There's no swirls and convection, anything like that. And you can go very infinitesimally slowly up. You can expand. You can go infinitesimally slow down. And he says, and this is going to be the closest thing to frictionless as you can get. All right? So he conjectured that perhaps he used the motive power of heat. It should be ultimately independent of the agents employed. Instead of water and steam, it could be anything. So he just idealized some gas. And he talked about the transfer of caloric. The caloric was this heat energy, and how you can turn heat energy into mechanical energy. All right. So that was Carnot. He died very young, in his 30s. And fast forward lots of time later, and in 1854, Clausius found that this caloric, he said that this mysterious amount of heat on the temperature T1 and T2, T1 is hot temperature, T1 is cold temperature, and that he found out that if you take this integral of the Q over T, the transfer of heat over the temperature, and you go up and let the piston expand, and then you go to a cold thing and let the piston contract, that the round trip becomes 0 for this idealized, super slow, frictionless thing. So it began to look like a potential energy term. Okay? And so if it's a reversible engine going infinitesimally slowly up and down, frictionless, it looks like 0 energy in a round trip loop. And anything else you would have something different. You would lose energy. So he defined this funny thing. So the integral of the change in heat over temperature going up the path and back down the path is, he said, that motive power of heat was the efficiency of a reversible engine. And what Clausius proved was the efficiency of this heat engine could at best be the temperature at hot minus temperature of cold over temperature of hot. No matter how smart you are, that's the limit. You want to make a better heat engine, you work at a higher temperature and you dump the energy at a colder temperature. And that was it. It's a beautiful law, no matter how smart you are. It's like the uncertainty principle of Heisenberg. No matter how smart you are, if you want to measure position and momentum at the same time, this is as good as it will be. Big greater than equal, essentially Planck's constant, plus factors of 2 pi. Okay, so that was Carnot. Now, that was the definition of entropy. And when you learned entropy, when I learned entropy as an undergraduate using this definition, I didn't understand it. Not many people understood it. And even beyond that, this is a really weird thing. And in a standard physics textbook by Cattell and Romer, they said the utility of the conception of entropy is limited by the fact that it does not correspond directly to any directly measurable physical property, but is merely a mathematical function of the definition of absolute temperature. Why absolute temperature? Because if you have your thing and you get heat from a hot reservoir, and then you put it at 0 Kelvin and get rid of the waste heat, then you can get an efficiency of 1. And that was that perfect gas law analysis of the Carnot cycle that actually defined what temperature really was in absolute value terms. It was not named after Clausius, it was named after Kelvin due to complicated reasons. But anyway, they both made very great contributions. But actually, Cattell and Romer are wrong. We can actually measure entropy directly. And so for that, we need another definition of temperature, not as this funny integral. And so for that, we need Boltzmann's contribution. So this is Ludwig Boltzmann. And on his tombstone was that S equals a physical constant, now called Boltzmann's constant, times the log of W, where W is the number of accessible states. Okay, so that's the definition of entropy. And so what does that mean? So I'm going to give you a very simple example. Suppose you have, and this was considered in the 30s or 40s by Landauer, and Landauer asked the question, if you have some memory and you wanted to read out the memory and then reset it back for another memory, what's the minimum energy that you need if this computer is going to work at a certain temperature T? Okay? During that time, Landauer also showed that you can actually do things on the actual computation if you do it in a kind of Carnot sort of way, very slowly, reversibly, that you actually don't need energy to do the computation, and it was only the memory. Okay, so what does that mean? Remember the definition of entropy, K log 2. Suppose you have a memory, it could be a 1 and a 0. So log 2 is, there's two accessible states. So in one state where you're about to read out, you don't know what it is, so the entropy is K log 2. After you've read it out, you know what state it is, so there's only one accessible state. So it's K log 1. K log 1 is 0, so the difference in entropy is K log 2. If you assume the second law of thermodynamics. Now, Landauer was actually trying to be more ambitious, and he was trying to show from even more first principles than the second law that this was the limit for computation memory. Now, let me also pause and say that this idea of having a memory readout of K log 2 is irreversible because you've done something, you've read out some knowledge. And except for perhaps the previous president of the United States, once you actually read out some data, that's it. Sorry. That person thought it was reversible. So anyway, so that's where K log 2 comes from. And so there's been some measurements about this. And this is taken from an article by Jeff Boker in Science Advances. Jeff is a professor in the electrical engineering department at Berkeley. When I was at Bell Labs, I was his department head, which meant I worked for him. I made sure he was happy and could do the research he wanted to do. So it's good to see this. And so what is that dotted line? That's the Landauer limit. If you go to higher and higher temperature, K log 2 increases. And the red line is the average of what it took to read out a spin and flip it back to reset it. So within 20%, he's getting K log 2, which means within 20%, he's reading out entropy, the minimum quantized entropy, which is really cool. Okay? So it's not only, no matter how smart you are, you can actually design things to really adiabatically change that spin to do it. Okay, so let's move on. All right, so it's not, what drives life and chemical reactions and everything is not just the entropy, but it's Gibbs showed that it's of what is called the Gibbs free energy, which is the enthalpy. Delta H is the actual net energy you have to introduce to change a molecular state or something, minus T times the log of entropy. So here's a little quiz. This is a polymer, and this is one accessible state of the polymer. And here's another, same polymer, it's a little crumpled up. And finally, another polymer, same polymer, is even more crumpled. And so the question is, which state is more equally likely? And the answer in statistical physics is each is equally likely. However, there are many more ways of arranging the polymer to be a little squiggly thing than a straight out thing. A straight out thing, that's it. It's this way, this way, this way. But a squiggly thing, many, many ways. So when you stretch out a polymer, it will contract like a rubber band because that's how rubber bands work. You're stretching out polymers, and then wants to go back to a much more disordered state. Entropy is increasing. And so we actually did this experiment where we stretched out a single molecule of DNA and let it go. And lo and behold, it collapsed like a rubber band. And we can test some basic fundamental theories of polymer physics this way, but it's real life. It's in an environment, it's only entropy that's making it shrink. Okay. All right. So the second law describes the properties of large numbers, and that's why the second law of thermodynamics, it was considered sacred, because general relativity may come and go, quantum mechanics may come and go, string theory may certainly come and go, but whether the laws of mathematics will come and go may not. Now, as you say, of course, mathematics will never be, because after all, God came down from a mountain and told us the laws of mathematics. Not. And there's one of my favorite movies is Abraham Lincoln Spielberg, where this is Abraham Lincoln. He's talking to two young telegraph guys, and he's deciding whether there's some emissary from the South. They want to, maybe seek peace, but the deal would be the 13th Amendment would have to go. And so Lincoln is telling these two young kids, when he was a young man, when Lincoln was a young man, he read actually the works by Euclid. He was really self-taught, but not only self-taught, he found that he loved the logic of Euclid. And he says in the movie, things that are equal to the same thing are equal to each other. That's a rule of mathematical reasoning. It's true because it works. Laws of logic are experimental. Okay? As far as I know, no Moses, no tablets. Okay? But anyway, think about it. So let me tell you about new experimental theoretical advances in entropy. And I'm going to start by talking about molecular motors. But again, since many of you don't know much biology, I'm going to start by showing you a little movie put together by Ron Vale, who is a very eminent biologist who discovered kinesin and did a lot of seminal work in the motor. I'm going to talk about Dynein.
Just like a busy city, there's constant motion inside of your cells. There's new construction, demolition, and most importantly, transporting goods from one place in the cell to another. Cells transport goods along cellular roadways. To transport cargo along these roads, the cells use motor proteins. Kinesin is one of these motor proteins. If you didn't have kinesin and other motor proteins, you simply wouldn't be alive. All the cells in your body depend upon these tiny motor proteins to organize and power themselves, to divide and multiply, and to communicate with other cells. Let's start with how you began, as a fertilized egg. That egg has to divide into many more cells. All of those divisions require kinesin and many other motor proteins. Development, forming tissues and different parts of your body, requires molecular motion and motor proteins. Every cell of your body requires them for survival.
Okay, so that's an introduction to motor proteins. This is an introduction to how motor proteins don't work. They are not these really well-behaved little macroscopic machines that go like this, because these motor proteins are embedded in fluid at body temperature, and there's a lot of jiggling around, okay? So that image is a not so good one. So what we're going to do is we're going to talk about motor proteins and that transport chemical messages between inside a cell, and the cells we're going to look at are neurons. Neurons are really having a special disadvantage because the body of the cell of a neuron, if it, let's say a dorsal ganglion root DRG neuron, could be over here, and the axons could be over. The axon is a long, skinny thing, and then the rest of it can be way over there. How long a distance? Well, a single neuron can go from your hip to your knee, one cell. It's not atypical to have them go hundreds of centimeters to a millimeter in your spinal cord. And so what happens is that if this is the distal axon at the end on the right, the other neurons will send chemical messages to this neuron saying, you know, I'm talking to you, we're okay, we're shaking hands, it's all good. How does the body of the cell know that it's all good? Those molecules get absorbed by the axon into a little vesicle called an endosome in this cartoon, and they package these motors called Dynein onto them and traffic it back to the cell. So the reason you can get this thing is there's actually a motor that burns units of energy that actually walk back to the cell because diffusion won't transport that long a distance. Kinesin goes the other way. Very essential stuff like, for example, mitochondria that makes the ATP energy that the cell uses. It needs energy at the tip, and so that all gets transported. And so the mitochondria get transported that way, but then you got to bring the kinesin back, so it's got to be transported. Okay. The motor I'm going to talk about is called Dynein, and so it's a complicated motor. It's 1.5 megadaltons. 1 dalton is 1 hydrogen unit, so 1.5 million hydrogen units big. It's an amino acid chain that's a huge, humongous long chain, and the molecule is just the light and blue guys. But in addition to that, there's all sorts of accessory proteins that attach the dyneins to each other, and finally to this endosome, which is carrying these messengers. Okay, so despite the fact that dynein was discovered decades ago, its full operation is still not known. And this is a picture of a cellular picture of what one of those monomers will look like. And so now when I show this to some biologists and they look at something like this, it's sort of like Snoopy hearing Charlie Brown talk, and what Snoopy hears is wah, 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 wah. Okay, so so for you physicists in the audience, what these are descriptions, and this little thing that goes like this is polypeptide chains that curl around in sort of a helix form. And these little blobs here, each of these little blobs would be little amino acids, okay? So this whole thing is 150 million hydrogen masses worth of strung together amino acids. It's weird because this thing, which has these 6 domains in these different colors, they're all different. The motor itself doesn't act like kinesin, it doesn't act like myosin. So those are motors that make your heart bump, muscles contract, and the thing that, you know, that cartoon that was going like that. This thing has nothing to do with those. It comes from a motor that's used to package DNA into viruses and motors that are used to unravel things. But there's a mystery as to why nature decided to use this class of structure to make this motor. But it's the only motor that works really effective in going, quote, backwards. Okay. Anyway. So this is a cartoon, a better cartoon of what a motor might look like. These are two monomers attached to some cargo back up here on the upper right. And you see it's kind of wiggling around. You see Brownian motion, and see it's deciding whether to step or not. And so it's kind of, finally decides it's going to step. And, its foot is going like this, and then it goes like that. And then this linker arm is in two states. It's in this straight yellow state where the linker is straight and everything. So that's a cartoon, again, due to Ronvale, of what it might really look like. Okay, so we're going to try and unpackage some of that. How are we going to do it? Well, we're going to follow the words of the great American philosopher of the 20th century. Here's Yogi Berra in a philosophical conversation. He was catcher for the New York Yankees. He said many, many wise things. And particularly, he said you can observe a lot by just watching. Like if you come to a fork in the road, take it. And two, you can observe a lot by just watching. I would say you can observe even more by watching for a long period of time. I also said something as I get older. I said, you have to go to your friends' memorial services, because if you don't go to theirs, they won't go to yours. Okay. Anyway, so we're going to use new probes, optical probes called upconverting nanoparticles. And the way these things work is you shine in infrared light, and it's a two-step excitation, and in the energy structures of rare earth ions, you can make two steps. So infrared light goes in, visible light comes out. They're good because the infrared light doesn't damage cells. They last for a very long time. So for many, many hours at the highest intensity, the quantum dots are in gray. They blink and they do things. They finally photobleach. And then organic dyes are getting better and better and better, but they too photobleach in tens of seconds, maybe a minute. And then the so-called fluorescent proteins photobleach in a few seconds. So those are all good things. When we started this, my idea was I didn't know anything about nanoparticles, so I'm going to work with the best nanoparticle people. They'll supply me with the particles. I know what biology I wanted to do. And the canonical wisdom is that you have a nanoparticle. The base is sodium yttrium fluoride, but that's not optically active. You put in a rare earth that has optical sensitivity, and you transfer the energy to erbium. And so only 20%, because they didn't know how to grow crystals that are better. But we figured out that we could grow crystals. So they gave us the particles, and we found out they were not as good as advertised because the physical characterization was through a scanning electron microscope. They looked beautiful. But when they did the optical characterization, they had to make them water soluble. They stick them in a cuvette, and they looked at the optical properties. But the particles clustered together, and for energy transfer reasons, they were brighter than expected. So it was all over the map. And so it took us a year to find out the particles all clustered. Once we figured out they were clustered, we had to figure out a way of knowing when they were not clustered. Once you know the way they're not clustered, you can figure out a way to keep them from clustering. So we started doing that, and in the process, we said, well, maybe we should think about making them ourselves. We found that you can go up by a factor of roughly, instead of 20% of YB, you can go to 90% YB. So it makes it 4 times better, 4.5 times better. Taking out the organic solvents to make them water soluble and putting on something that made them water soluble damaged the outside. We found that by accident by looking at our own SEM micrographs and TM micrographs, and there were little cracks. So we said, this is bad. So we figured out a way. We don't have to take out the organic molecules. We just lay over something that's water soluble, and you're only adding a fraction of a nanometer, a small fraction of a nanometer. So it didn't matter. So the result of that is that at intensities that use for animal imaging, there are 150 now, 200 times brighter. And at the high intensities we use for the high resolution stuff, they're still 6 to 10 times brighter. And we also found out how we can make them really small, down to 10 nanometers, and still have the volume brightness. The brightness depends on the number of rare earths. The transition is an inner shell transition, so it's not affected by any surface effect. And so that's the good thing. Our experiment was we would put immature neurons in a flow cell. We give it some chemical signals so that they grow their axons through the flow cell, 900 microns of gap. And then the idea was we would put our probes coated with a chemical signal. Remember, this neuron wants to grab the chemical signal and transport it back to the cell body to say, I'm fine, I'm happy. So if you put those molecules, or molecules like that, nerve growth factor, but we found a much cheaper substitute, onto them at the distal axon, they will scarf it up, put all the motor molecules on it that are needed, and just start transporting them back. So this is, and it would go and they would start walking backward towards the cell body, and we would track it over this 900 microns distance, and we could look for tens of minutes without killing the cell. And so this is a top view of what it looks like. This is a motion picture. Those are seconds reeling off. There is no background because we're exciting with infrared light. So because it's infrared light, there's no reactive ion species, so no bad photophysics stuff is being done either. And sometimes you saw that the motor might go backward. We think it's on the same track, so kinesin is taking over for reasons we don't understand. Sometimes it goes backward. These are rare events that go backward on a different track. Okay, so that's the raw data. So now we're going to say, okay, what do we do with the raw data? Because we now have tens of minutes of a single vesicle plodding along over a millimeter. And so that's distance versus time. And we're going to look at the segments that are roughly constant velocity. And what we're going to say is, okay, at time t equals t, it's over here, and a little time later, tau, it's over here. And we're going to look on all these red sections, and we're going to say, okay, we can concatenate all these journeys together. It's the same vesicle. And so if it's here at position 0 at time t equals 0 after 10 milliseconds, this histogram gives you the distribution after 40 milliseconds, after 100 milliseconds. And so the average is going forward. Sometimes it steps backward, but it's mostly going forward. And we're going to look at this, and this fits a Gaussian very well, so it's just empirical so far. So it's a Gaussian, and a Gaussian can be described by a mean of the Gaussian after a certain time, tau, and also the spread, the dispersion. And so we're going to plot twice the mean divided by the dispersion. You'll see 2 in about 30 minutes why it's 2, but just mean over dispersion. And lo and behold, we find for that one vesicle, and all these segments where it's constant velocity, there are many, many constant velocity segments. In that one vesicle, it would asymptote to some level over here. The dimensions is inverse nanometers. Why? Because it's mean divided by dispersion, distance over distance squared, so it's inverse nanometers. Sometimes it's 0.1, 0.2, 0.3. Okay? Sometimes, 40% of the time, it doesn't really reach steady state. We're not going to do that. That's much more complicated. So we're just going to look at the steady state. These are 12 neurons. They're all different. Some neurons reach only 2 values. Over here, this one, 2 values over here. Some reach 3. Some reach 5. And everything in between. Okay? So each of those boxes is one vesicle marching along for 900 microns. If you plot out these asymptotic values, you find that they're amazingly quantized, even though the vesicles themselves could be of different sizes and they have a different number of quantizations. Okay, so the question is, what's going on? Now, there's a clue, a suspicion that maybe this could be the number of motors moving the vesicle. We know this from electron micrograph images, where here's the vesicle, this is the axon, the railroad track, and these were what in 1865 were called cross bridges. And retrograde transfer means it's going backward to the cell body. Anterograde means it's going forward. That's kinesin. So retrograde, we don't see no motors, because we're not going to see motion. So 1 motor, 2 motors, 3 motors, 4 motors, 5 motors. And so it's, roughly looks like the same. So maybe it's the number of motors. Okay. So let's go with that. Number of motors, 1 motor, 2 motors, 3 motors. That's the quantization. All right, so now I put on my physics hat, or math hat, and let's say if you have 1 motor and it's trucking forward, and let's say after a certain time 1 motor has this Gaussian spread and dispersion, and you have another motor, but the other motor is not talking to the first motor. They're both connected to the vesicle, but they're not really talking to each other because they're just getting their fuel. And so if that's true, then the displacement of motor 1 with probability P1 and displacement of motor 2 with probability T2, the displacements are x1 and x2. They're independent variables. So what that probability tells us is that if they're independent motors, the combined effect would be the product of the 2 probability functions. That's just math. Well, if there are Gaussians, this is good. We know that the product of 2 Gaussians are Gaussian. And if they're independent same motors, they have same displacement and spread, you can show analytically that the average speed is the same, but the dispersion goes down. Now, that might seem a little counterintuitive at first. And so the secret is you find out what the answer is, and you build up your intuition. OK? So I'm serious. It works every time. And so suppose you have 2 motors. And this is 1 motor, and this one goes like this. OK? This one hasn't moved. Went half as far. This one goes like this. This one hasn't moved. Went half as far. But you go twice as fast. OK? But you're going twice as fast. The displacement's the same. But you're making little steps. So the dispersion goes down. Suppose you have not 2 motors, but n motors. Lots of little steps. In the limit of lots of little steps, displacement the same, very steady motion. Dispersion goes even smaller. Okay? So that's the intuition. That's the intuition of the math I just showed you. So for n motors, the width is the same. We submitted this to a paper, submitted it to Science. The referee said, "You haven't proved this." Never mind. No, that's a proof. If then. If they're independent motors, then. But that's just math. What about biology? So it turns out, we looked back in the literature, and there was a nice experiment done, not for this reason, but it was done in 2012 where they looked at 1 Dynein motor, and they stick it on a microtubule. So the great thing about these so-called in vitro, in glass experiments, you know exactly what you put there. So they watched this motor walk, and they measured the speed. And then they put on what's called DNA origami. So each of those little circles is a double helix strand, and they link them together so it makes a kind of a rigid little thingy. And with DNA, you can play all sorts of tricks. So you can hang on motors at precisely where you want them. So you have this DNA origami. You have 1 motor in 1D. You have 2 motors in 2D. 4D, you have 4 motors, and so on. And lo and behold, what they found is that the velocity was the same. As you get to too many motors, the density was getting in the way, and so they might have been interfering. But for 1, 2, and 3 motors, it's fine. This is a perfect mathematical realization of independent motors. Okay? Now, in our real live cell experiments, they're kind of attached to the same vesicle, not quite the same, so there's maybe a 5% change when it goes. Okay? So for that, we say, okay, we know how many motors there are. It seems to fit the mathematical model, and the in vitro experiments are good. Great. All right. Now we can do some simple math. We know how many motors there are in this live cell. We know that these motors are burning units of energy called ATP, adenosine triphosphate. And they're working at, let's say, 37 degrees body temperature. And so the amount of free energy, you start with adenosine triphosphate, you break a phosphate bond, and you're left with ADP and a phosphate. So there's more entropy in that, and you're breaking a chemical bond. So you can calculate what the free energy release is, and it turns out to be 12 kT if you're working at 37 degrees. You watch how this motor goes, and you look at the fastest motor, that's a single motor, going at 6 microns a second, really tooling along. And you say, well, we know what the viscous drag is, because we know the viscosity. How do you know the viscosity? Well, you can put in a cell a particle the same size as the vesicle and see how it diffuses. Now, the viscosity is dependent on the size of the molecule. If it's a small little itty-bitty dye molecule, the viscosity is close to water. But if it's a big thing like 100 nanometer radius, the viscosity is 1,000 times bigger. So it depends on how big it is. But luckily, we have these measurements. And we find that as the motor is trucking along, it's at least 50% efficient. If it's burning 2 ATPs, it would be 90% efficient if it's burning 1 ATP. Okay? And so we'll get to, because the accepted model at this time was 1 ATP. Okay. All right. Let's continue. The particles are pretty good, and we can make them pretty big. And so we can measure how they're moving the vesicles at body temperature 37 degrees, at 30 degrees, 22 degrees. And we begin to see individual single steps. So that's actually pretty good, because never before could you actually track single steps for minutes or even seconds without frying the cell. But we can see for 20 minutes single molecular steps for a very long time. So that, because our particles are really good particles. And so these histograms are the steps size distribution. How do we know the step size distribution? Because you see it's pretty noisy. So you just have a maximum likelihood program that decides when it takes a step and how big is the step. And so how big the step is, we can form a histogram, 22 degrees, 30 degrees, 37 degrees. We showed this data to Ron Vale 3 years ago. He said, wow, at body temperature, there doesn't take backward steps. Well, that's not quite true, because we're drawing a Gaussian over here, and we're pretending there's something here. We're not pretending. We know, because of the noise, we can't see 4 nanometer steps. We certainly can't see 0 nanometer steps, or -4. So it's hidden in the noise. But the good news is, if you compare it to the in vitro experiment. So the in vitro guys went to really low ATP concentration. How low? 3 orders of magnitude lower, to slow it up so they could see single molecular steps with the same resolution as we could see. Okay? 3 orders of magnitude. You're supposed to say, wow. Anyway, this is Ron Vale's data, and then later data by Ahmed Yildiz, who was a postdoc with Ron Vale at the time. Also, they can lay right on top of each other. So the good news is we know we're looking at Dynein. To the in vitro guys, they've assembled enough of the parts that it's beginning to act like it was in a live cell. Okay, so that's good. Since we can measure how big the steps are, we can also measure how long it takes to go to the next step. It's called the dwell time. And when you do that, you find, lo and behold, you get these histogram distributions. It's a rise and a fall. The solid line is a fit to a particular model. The model is very simple. If this motor was just a simple rate constant, it should be a single exponential decay. Think of an atom in an excited state. Its probability of de-excitation is independent of time, and so you can show that it's a single exponential decay. If you see a rise and a fall, there's a hint that there may be two rate constants. If there are two rate constants, you do, in that diagram in the upper right, red is rate constant, and it happens at some time t1, and then you convolute that with t2. So the convolution of these two single exponential decays gives you a function, and it turns out that this convolution function, if the rate constants are equal, you can get these black line fits. So what it says is, in this new regime of 3 orders of magnitude higher ATP concentration, we see two rate constants, not one. And so now the earlier data of 2006 only saw one rate constant, but we're going to get to that in a moment. OK, so the unit of fuel is ATP. So ATP lands on one of these active sites in the Dynein. I'm not going to go into that, but just say it goes from an empty site to ATP. As soon as ATP lands, the molecule kind of closes around it and forms these chemical links, and then electron transfer occurs and breaks the phosphate bond. That's really fast. That's way sub-millisecond. OK? Then now you've got ATP is turned into ADP plus PI, but it's still on the molecule. And the next thing that happens is the phosphate group gets released. PI is phosphate group, and it has ADP. And the next thing after that is ADP leaves, and it's empty, and it goes into a full cycle. So we looked at all the rates that were measured in this molecule, Dynein. APO to ATP, well, at our concentrations, it occurs in a millisecond, roughly. It's measured rates. ATP to phosphate burning, sub-millisecond, really fast. ADP to APO was measured kinetically by another group years ago, and it was about a millisecond at room temperature, much faster at 37 degrees. So you look at all those rates, there's only one left, phosphate. Has not been measured. But there are two. So it has to be two phosphates. OK? So all of a sudden, we're faced with this dilemma. But the prevailing model says only one ATP burning, and this one says got to be two, and we has to be the phosphate release. The prevailing model was built on data where the on rate of ATP was the rate limiting step. All these other things were much faster. So by going in 3 orders of magnitude different regime, we're forced to confront this. But then, I'm not going to go into the details of the model, but I've just suffice it to say that, when did I start? At 4:35? No. Anyway, suffice it to say that what if our model was right and you're in the rate limiting step where it's on rate of ATP, but there are two sites? Then shouldn't have those guys seen a rise and a fall, right? It's just in a different regime, so a different thing. And so we went back and talked more with Ron Vale and Stefan Niekamp and said, well, there's a little hint of this. But maybe you shouldn't histogram it. Just use what we did, maximum likelihood, find out when it made the transition, don't presume anything, and then plot that as just discrete units. So histogramming makes some boundaries. And so Ron says, yeah, we think that's a better way of doing it. We'll check out, see if your program works. Stefan Niekamp will do it. And then he writes back and says, oops, two time constants, they're equal. Great. Now, I was a little nervous by that little gaps, but we show by simulating two time constants are equal with their time resolution of their experiment, that's a sampling thing. Okay, so I said, that's good. Okay, so we then started work with Ahmed and Yildiz and say, hey, we got this. And so we're trying to sort that out. But so we're thinking it was probably there for a decade or two. But there was too much expectation it was 1 ATP for other reasons. For example, if it's 2 ATPs, then if you look at concentration of ATP and it rises, there should be a sigmoidal dependence. If it's 1 ATP, there's something called the Hill coefficient. For those of you who know molecular biology, kinetics, Hill coefficient 1 active site, Hill coefficient 2. And so they found Hill coefficient 1, except they made a mistake. Hill coefficient 1, two sites can have Hill coefficient 1 if they're separated by irreversible event. There's nothing more irreversible than burning ATP. Therefore, you can't tell from Hill coefficient whether it's 1 or 2. And so we went through everything that said it had to be 1 and found that those reasons disappeared. Okay, but it's cooler than that because you can actually, the dwell time, so this is the dispersion, T is the time between steps. So it's the dispersion in steps normalized by the time quantity squared. So it's the time in between steps. Remember, we were in two different regimes. In the in vitro guys, dominated by ATP on rate. In our, say, dominated by PI release. What if you adjusted the ATP concentration so the on rate is equal to the off rate and there are 4 steps? Then you can show mathematically that you should peak at n equals 4. But we were in the limits in both of those, and so we proposed in the paper that was rejected, still trying to get published, that if you do this experiment, you can see n equals 4 specifically in, and it would be different in different experiments because the off rate would be different. These are the fits to the Hill coefficient for 2 ATPs of the data that was actually taken by Ahmed Yildiz. So the good news is Ahmed will be sending us his molecular constructs of 2 dyneins and attached to a dynactin. We'll put our probe on it and we'll see what happens. So we'll be working with Ahmed and trying to figure this out. Ron Vale was very cute. He's now the head of Genelia Farms. And he said, oh, shucks, I really want to do this experiment. So I said, Ron, why don't you just sneak out just because you're head of this couple $100 million a year organization? It doesn't mean you're the boss. You can sneak out. But never mind. OK, so let me tell you about a simple model and what the diffusion is. OK, so that's a bunch of math. I'm going to do this. OK, so here's the simple model. They have 2 steps, 2 legs of the dynein monomer. And remember that cartoon where it said, I'm wiggling around, wiggling around, wiggling around. What really happens is I'm wiggling around due to diffusion. I'm mostly down here. I'm up here only for a short bit of time. I wiggle around, I go down, I wiggle around, I go down. And finally, PI leaves. And we know from previous experiments it does a power stroke. So we know those things. We also know more. These structures of that, remember that yellow thing in that cartoon was the linker arm, and there are 2 structures. It's in this bent thing, and we know these angles, they've been measured. We know the size of the molecule, that's been measured, and we know these distances. So what do we say? We say, well, we make a thing. We have these 2 feet, and ATP, now it's wiggling around, it's in a different structure. It's forward biased a little bit. So it does a random walk, and finally it walks. How long does it does a random walk? We know the viscosity. We know the time it's up. And for higher temperatures, the PI leaves sooner. We know how much faster it goes. So we stick all that stuff in, and we say, this thing is wiggling around. It's overdamped. It's attached by an effective spring constant to the rest of the thing. So the spring constant is F equals kappa x. Drag force is viscous drag. You solve that equation, and it's a very simple equation. For very short times, it looks like this thing is just simply a random walk diffusion. For very long times, it's testing anywhere it can go by the spring constant. And we find in this very simple model that we're only solving for one spring constant. It fits the three temperatures beautifully. And so it's like very, very cool. All right. So it works. Diffusion works. Viscosity works. We now know it's spending most of its time here and it's going up and down. But as a physicist, I'm saying, let's really test it. Let's plug in all the numbers and see. And that works too. And it works. And to within 30% of the parameters in the cell biology experiments that people have done, it fits. OK, so let me switch gears and step back a little and talk about, is life based on the laws of physics? And that's a question posed by Erwin Schrödinger in his lectures in Dublin in 1944. And he says, from all we've learned from the structure of living matter, we must be prepared to find it working in a manner that cannot be reduced to ordinary laws of physics, not because there's any new force, but because the construction is different from anything we've tested in the physical laboratory. OK, if Newton was the size of a bacteria, but with the brains of Newton, his laws would be very different. There is no inertia. As soon as you stop pulling or pushing, everything comes to a screeching halt. It's like bureaucracy. An object with no net motion has this Brownian motion. That's really weird, you know, like the hand of God shaking you. And so F does not equal ma. To repeat point one again, is force is proportional to velocity. And in living organisms, they're embedded in this overdamped, viscous, jiggling stuff. So how does it work? And the beauty of how it works is that it does work. Don't have time. In my next hour, I can tell you how beautifully this motor uses fluctuations, but I want to get to physics. OK. That's my 30-minute warning. Suppose you have two molecules that come and they collide with each other. So time going up. And if you reverse the movie, time going down, you can't tell whether it's going up or down. Not a problem. OK, do something different. Drop some blue molecules into a beaker of clear water molecules, and you can tell what's happening. Each of the blue molecules looks around and says, hey, I can go anywhere in the beaker I want. And so when it reaches equilibrium, I can be anywhere. So the number of accessible states of those blue molecules has increased from the little droplet to big thing. So entropy has just increased. Okay? There's a new theorem that was discovered in the 1990s. You think everything was done in the 1880s? No, 1990s. And it quantified this notion of those blue molecules going anywhere, and said the probability of entropy increasing, the entropy is increasing because the blue molecules can go anywhere. And so the probability of the movie going forward in time divided by the probability of moving going backward in time is not infinity. It's e to the change in the entropy over Boltzmann's constant. That means for very, very short amounts of time, it can go backward in time. Okay? It has to, because the collision laws are time reversal invariant. So it's saying it's the laws of large numbers that make it go forward in time, but it puts it on a very quantitative basis, exactly how it goes forward in time. So this is called the fluctuation theorem, as opposed to the fluctuation dissipation theorem. It was discovered in a computer simulation in 93, proven in 1994. It doesn't violate the second law. It just says, in the second law, you wait a long time, and so the unlikely is so exponentially unlikely, forget about it. And the amazing thing about the fluctuation theorem is it works in microscopic, micron-sized particles. person did an experiment. They dropped a bead in air, get rid of all the air currents, and watched it fall due to gravity. And it falls upward for short periods of time because the fluctuations make it go up. And it quantitatively agrees with the fluctuation theorem. Okay? But of course, you wait long enough, it's only falling down. So the great thing about the fluctuation theorem, it doesn't have to be in thermal equilibrium. It can be anything. It's just about laws of large numbers. So now you're liberated from that. Okay, probability going forward in time, going backward in time is proportional to the probability of stepping forward in time forward, those measurements, versus backward. And so you consider two paths. You start at time t equals 0, you're at that origin. One trajectory gives you the red line, another trajectory gives you a blue line. What's the time reverse thing? The dotted lines. But you want to start t equals 0, so you just go like that. So going backward in time is just an algebraic construction once you know what's happening going forward in time. Okay. All right. Well, we know what the probability is going forward in time. It's our friend the Gaussian. And so if we have our friend the Gaussian, we look at that. Going forward in time versus going backward in time is the Gaussians. But the Gaussians are really cool because if I take the logarithm of p of delta x and p of reverse time minus delta x, that means that I'm adding and subtracting the exponents, and that even I can do. And so when you take the log of that, you find that delta S, the change in entropy, is equal to our friend phi, twice the displacement over the dispersion. Okay? Except now it's different. Our phi before was just didn't matter what it was, but now we associate with a thermodynamic entity, entropy. And when we have entropy, we can start writing down. We start with thermodynamic things, but it doesn't matter because this is fluctuation theorem doesn't need equilibrium thermodynamics. So the change in entropy is the enthalpic energy times the work done. The work could be moving the thing through a viscous medium plus all the internal friction. When you're going a full cycle, the enthalpic energy, you start with ATP, you burn it, you start again with ATP. So that turns out to be 0. And so that delta Q then is simply this. So the change in entropy is related to the work divided by the temperature. And that, for thermodynamics, but we'll see in a second. But anyway, and so then you have work as force times distance, but you get rid of the delta x more algebra. Okay. If I calculate what the force, drag force would be based on equilibrium thermodynamics, I get to assign a temperature, 310 Kelvin, and I find that this measurement gives us the force is 0.43 piconewtons. If I say, but I know the size of the vesicle, I know the viscosity of the vesicle, and I know the velocity, therefore I know the work. What's that force? It's 2.5 piconewtons. Whereas this other thing was an upper limit because it's also the internal friction. Okay, so this doesn't match. So what could be wrong? Well, let's suspend belief for a little while and say, well, the only other thing is the temperature. What if the temperature is not the cell temperature? What if the temperature is 5.7 times the cell temperature? Then it matches. Okay. Yeah, sure, you say. 5.7 times 3 times Kelvin is, everything gets ionized, but no. Not true. So let me talk about non-equilibrium thermodynamics. So way back when, in 1931, Lars Ansager said, if I take a system and it's in equilibrium, we know that the fluctuations about equilibrium are related to the dissipation, the drag, the Einstein relationship, Brownian motion and viscous drag. That's the fluctuation dissipation theorem. Suppose I take this and put a little bit out of equilibrium. Its approach back to equilibrium should be related to fluctuations about equilibrium. That was a bold conjecture because no one thought about non-equilibrium. But in hindsight, after 50 or 60 years, or 31 to 2012, okay, you can't tell if the system were put out of thermal equilibrium by some unknown force, or there's a big statistical fluctuation to put it there, and then watch it come back. Got to be the same. So the Onsager regression hypothesis was the first attempt to study quantitative non-equilibrium thermodynamics, and for that, 30 years later, he got a Nobel Prize. Okay. I'm going to skip the fancy math because I'm just a simple experimentalist. And I'm going to tell you, I spent 3 years understanding the fancy math and then tried to figure out what's really happening. So what's really happening? You go to Langevin equation. It's F equals ma. Mass times the change of velocity. Now there are two forces. There's the motor force that's pushing along, and there's the viscous drag force, and then there's this Brownian motion term, okay? Psi. Now the Brownian motion term has no really fast correlations, and so, but now the motor is stepping really rapidly, but it's not a really ordered step. It decides when it steps, and sometimes it steps backwards, and so there are fluctuations. So I say, okay, let's go to the center of mass of the vesicle. Center of mass of the vesicle, what do you see? You just see the motor fluctuations. It either steps half backward or half forward. You see the Brownian motion fluctuations, and you see the viscous drag of the center mass velocity. I look at that equation and say, I know how to solve that because, heck, I'm just going to imitate what happened for fluctuation dissipation theorem. The only difference is that you have now two fluctuating terms, but this one was a delta function. So what about that one? Well, it's stepping really fast, like every couple of milliseconds, and you look for a long enough period of time, so you're averaging over a couple of steps, then all of a sudden where the vesicle is at this point in time and where the vesicle is in this point in time are uncorrelated. And it's fluctuating. Uncorrelated fluctuation, equilibrium. Okay? So then you get a delta function, but with the proviso you have to average over a couple of steps. And then all of a sudden you have two terms. You have the normal Brownian motion, but you got this mother motor that's burning kT. And when it decides to step forward, it can make this humongous vesicle forward, and in center mass it's going like this. Okay? So if you do that, and you say, oh, in the center mass, you saw it, delta function, widening curve, widening curve, widening curve. Okay, that's random walk. Now I see a random walk. What's the temperature? 5.7 times 310. Water is not being vaporized and ionized. It's just the fluctuation of the vesicle because the motor is so powerful. That's what's really happening. At that point, I say, what's the big deal? Well, the big deal is this is out of thermal equilibrium, but it's a bigger deal, and I'm just going to in a moment. And the bigger deal is you go back to the fluctuation theorem, which I've said, and after many, many steps, you can look at many steps, and after many steps, it steps 1, P1, steps 2, P2. After many steps, you're doing the convolution of many steps. There's a theorem that says when you convolute any integrable function, I think it's a theorem, I couldn't find it, but I'm sure it's a function, because if I convolute many Gaussians, I get a Gaussian of a certain width and a certain thing. And the central limit theorem says when you convolute many things, you always get a Gaussian. So, so if anyone, mathematicians, then you point me to where it was proven. But anyway. Okay. So what this says is that if you convolute these stepping functions, and I take the normalized displacement, remember ns squared is the dispersion of this motor system, and mu is the displacement, and n is the number of steps, I get something remarkable. I can prove analytically, if this is a Gaussian, then the product of this dispersion, how orderly the motion is, times T times delta S, the minimum entropy needed to get this order has to be greater than or equal to 2 kT. It's an uncertainty principle, like delta x, delta p. If you have superorder, tick, 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 infinite energy. Really bad order, rate constant, single rate constant, clunk. Another one, clunk. Okay, then the dispersion turns out to be 1. Okay? And 2 motors, 2 energy is more ordered. The dispersion of 2-step is less than 1. N steps gets an hour. But of course, you're paying more energy, but you're burning 1 ATP, 2 ATPs, 3 ATPs. So this is trivial examples of this. So it's an uncertainty relationship. And in general, it's been proven that this is true. I was very proud of myself. It was 3 years ago, and I figured this out. And I said, wow, yeah, I'm a lot slower, but I can still do math. Because I can do integrals of Gaussians. Okay. But it turned out there were 2 other theorists who figured it out about the same time and published it 6 months after. So, oh well, it's okay. I'm an experimentalist. So anyway, this tells you what the minimum entropy is needed to do something in any precision or order. For example, it doesn't have to be biological. It can be a Mickey Mouse quartz watch, good to a second a day in accuracy, but we're talking about precision, which is different than accuracy. Accuracy is absolute value. It turns out Mickey Mouse watches are precise to 100 times more than that. And so that enables the number of joules. So, and this uncertainty limit is related to that Landauer limit. Okay? Because now we have from the Landauer limit, what's the minimum entropy to read out a memory bit? We now have what's the minimum entropy to do anything of any order in any system, whether it be a motor or whatever. Okay. Now, there's been criticism of Landauer and followed by Bennett. The criticism was not the result. The criticism, they might have been using circular reasoning, but now it's okay in the sense that it doesn't violate the second law, and from the second law, of course, it has to be true. And so now there's a realization that computation and biology and many other things we really care about are applying these Landauer limits, this entropy change. And in the days when Landauer and Bennett were doing their work, we didn't have an understanding of non-equilibrium thermodynamics that we have today. And so the irony is the things we really care about are actually out of equilibrium. And so now we have a language to do this. And so I think that's pretty cool. It was a happy accident that Dynein in a cell is out of equilibrium, because then I got to learn StatMech. All right, thank you. Was that understandable, at least partly? At least you laughed at the jokes. That's good. Okay. Okay. Yes?
This is a question maybe general, may not be general enough, but you're saying that I'm really impressed by the 3 orders of magnitude improvement of sensitivity. Is that a new approach, a new experimental method, or
That's
just the way that you do?
No, it's our particles. We got to make really good particles. The referees didn't point this out when they rejected our paper. In fact, one of them said, the data looks really quiet. Did you smooth it? And said, no, we're at 5 million counts a second, not 5,000.
Right, but when you say new particles, you mean the motor is different, or?
Our rare earth nanoparticles that we were synthesizing are much better. They're much better. Turns out we now know how to get these in the cytosol of cells. We think we can get them to nuclei. They don't photobleach. So we can do, I'm trying to impress upon you, the ability to track is really important. The ability to track for hours is going to be incredibly important. We only had to track for tens of minutes. You get a totally different view of nature when you can track. And so what we think is that it can open up lots of new biology so you can track for hours, days, and how the nucleus decides what
So
genes
your
to train.
nanoparticle is your sensor, basically. Wow.
Yeah. Yeah, we would have hoped one of the referees to say, these are really cool nanoparticles. I'm not bitter. Okay.
This might be wrong, but when you were doing ATP at the beginning, it says 12 kBT.
Yeah.
Is that 12 somehow related to the effective temperature?
No.
It's not.
No. So there's a natural, you can talk about, so this is free energy. You have, a molecule with a certain bond. You break the chemical bond, and you have two pieces. So there's increase in free energy because of entropy. And so it's based on the concentration of ATP in a cell and ADP and PI in a cell. And so that concentration determines what the net free energy is. And it's concentration dependent. If you go to a higher and higher concentration of ADP, there will be a time when it can go in the reverse direction. In fact, people have done this. They've taken a motor that's used to make ATP from ADP and PI based on a proton pump. So it actually turns it, squeezes the molecules together and makes ATP. But if you feed the motor ATP and don't let it turn on itself, it goes the other way and burns it. And so depending on the concentration of ADP and ATP, you can make it go either way. It's a chemical reaction. But it's 3 orders of magnitude more ATP than ADP, maintained by the cell. Okay, so we just take that as a given. Okay, but yeah, so it's just the energy that's available. Uh-oh. This is a bad sign. Okay. So you're either very tired, you didn't understand anything, or I just want to remind you of George Schultz's pig pen. This is us humans. We're taking in all sorts of energy. We're the organisms decreasing the entropy. They're growing, they're learning, they're all good stuff. But the debris of entropy left behind is enormous. We are the world champions of the amount of entropy reproduced, and we humans emit 40 billion tons of carbon dioxide a year, including methane and N2O. It's equivalent to over 50 gigatons per year, and we also, by the way, the humans and the animals we eat are 95% of all mammal mass in the world. We've bioengineered the world. We've engineered the world. And we're emitting these number of gigatons equivalent per year. So this is incredibly efficient entropy generators. You should all be proud of yourselves. Not.
That's without computers.
What?
That's without computers and information.
Well, we now know if you do it right, it could be reversible. And if you're like Trump, you don't have to pay that entropy price, because it's reversible too. Anyway, so yes.
Suppose you could attach some active particles instead of recursive particles, what would happen if you could turn on the speed on all these molecular motors?
The speed is dictated by the PI release. You can up the temperature, but at a not much higher temperature, biology begins to fall apart. Okay, so and you can turn it down so it goes much slower. But this is not a practical machine. Oh, by the way, we don't know what the upper limit is of the machine. So that's another thing that was misunderstood because people would take optical tweezers and they would attach it to these motors in vitro, and you would pull backward, and they would find out at what point does the motor detach or stall. That is not the same, because think of this motor. It's foot in the air, it's going like this. So when it's both feet on the ground, that's when it actually pulls. If it's up here, it's detachable, right? And so that's not. And so people who use optical tweezers have to take the learner's manual to know what it's really measuring. But there is no driver test for optical tweezers. And so in any case, the motor going forward, it's a funny thing, because the faster it goes, the more efficient it is. Consider a 500 horsepower Ferrari going on an interstate at 50 miles an hour. There's no, with rock hard tires, there's no, this is a terrible motor. It's like few percent efficient. You only use the 500 horsepower when you're really accelerating. Okay, and so the same with so the efficiency of the motor is dependent on the load until it stops. So the right experiment is to increase the viscosity in the vitro until it stops. How do you do that? Well, you do it by imitating the cells. So you put in crowding, so the motor is like 200 nanometers, and it bumps into stuff, but you want to get the ATP in there. So it's possible to do that, but those experiments haven't been done. What's nice about coming out of Department of Energy is that all sorts of people waited for me to catch up again, which I'm very grateful. But so there's a whole bunch of experiments in vitro we're doing. We're talking to structural biologists. We have very detailed predictions about the detailed molecular structures, and so far in looking at the structures we can find in the so-called protein data banks, it's very consistent with our model, not with pre-existing models, but we're also trying to get them to do other controls, Angel Carter, for example, at Cambridge. And so it's going to be an exciting time. A whole bunch of people actually, when we talk to them, say, yeah, we'd love to, it's a falsifiable model. Nothing makes experimentalists happier than be able to test something that's falsifiable. Yep.
Maybe I understood a bit wrong, but so the idea of concept of like a very small scale to go with back in time, how does it relate with Heisenberg?
Doesn't. It's a Heisenberg principle in the sense that it's a product of two things has to be greater than or equal to something else. So in Heisenberg, the measurement of position and momentum has to be greater than or equal to Planck's constant over 2 pi. If it's Gaussian, it's the minimum. If it's the time it takes to make a measurement, delta t, and the precision of the measurement, delta e, has to be greater than or equal to Planck's constant. In this one, the minimum entropy cost energy you have to pay times the orderliness has to be greater than or equal to 2 kT. If the motor works at kT, that's the minimum. So it's an uncertainty principle in that sense.
Is it then possible that, like, if we have events like that, but time doesn't quantize as we think?
Say that again?
If we have events like that and measure things like that, is it possible that maybe time doesn't quantize as we think?
Oh, it's whether time is quantized. I'm not going to go into exotica like that. We're just saying after a certain amount of time, it's a theorem that's proved by Gavin Cooks that you have a certain change in entropy after this time. It's greater than or equal to 0. And after a certain time, and that's equal to the change, e to the change in entropy over t. We don't care if it's quantized. We're looking at such long periods of time, we'll never see the quantization anyway. Right? This is a statistical average. Okay.
So I'm afraid we have to wrap up. So I'd like to ask us all to give a big thank you to Professor Chu